we have our node cells or nodular cells, and we have our contractile cells. Okay. You guys need to know that your nodular cells, they're, again, the SA node, the AV node, all the fibers that conduct electrical impulses, that's what the nodal cells are. I'm referring to when I say nodal cells. We have other types of cells. We have the contractile cells. The contractile cells are the actual cells that contract and help with the depolarization, all that stuff that actually gets the heart muscle, the myocardium, to contract. So let's start off first by discussing how the nodal cell conducts an electrical impulse, its action potential, right? But I'm going to show you how it works in the cell and the influx and outflux of different ions. So first thing is the funny channels. The funny channels, what they do is they open up and they allow an influx of sodium. So sodium starts to go inside the cell, which is normally negatively charged, right? The resting membrane potential inside the cell, the intracellular resting membrane, is always negative, and the outside is always positive. But when the funny channels become activated, when they become activated, they start making the intracellular components more positive, right? So this becomes more positive, and of course the outside becomes more negative. So the sodium ions, as they start influxing inside the cell, they start activating the opening of other channels. First one, the other channel that we're going to be discussing is right now the T channels. And the T channels, what they do is they slowly allow calcium to enter the actual cell. Now, as the calcium is starting to come inside the cell, the positivity is increasing. And I'll show you how this looks like on the actual graph in a little bit with the action potential. But it becomes more positive now. Now, the T channels allow calcium to go in slowly, relatively slowly. But once it hits its threshold, that's when the L channels become activated, and they start bringing in a large amount of calcium, more calcium, and it generates a positive polarization of the cell. So now the nodal cells, the nodular cells, are positive inside. And when that happens, it starts to depolarize. It starts sending off electrical impulses. Well, because you have a lot of positive ions, your calcium ions, your cations, they start pushing the calcium through these gap junctions. And that's important for you guys to know. So the calcium goes to the gap junctions. And by the way, this part right here is the desmosome. And what they do is they maintain the integrity of the cell, and they allow all of them to stay in their anatomical location as they all contract, and they don't move around, unlike skeletal muscle. That's a little bit different, right? So you have the gap junctions, which are these ones right here, that allow the ions, the positive ions of calcium, to move over into the contractile cell. Now, remember, the resting membrane of all of these cells are negative usually. But in this particular case, as the ions start moving or shifting towards the contractile cell, they start making the cell more positive. And as they make it more positive, they start opening other channels. Initially, it opens up the voltage-gated sodium channel, and that allows sodium to enter the actual cell. So what's happening? It's becoming even more positive. So as it becomes more positive, it starts activating other channels, and it allows another calcium channel to open up as well. And so now you have more calcium going inside the cell, and that makes it even more positive. And that's where it reaches its threshold. And I'll show you what that looks like in just a bit. Now we have a situation here. We have a high concentration of positive ions or cations in the intracellular compartments, right? This does a few things. As that positive cation, the calcium, enters a cell, it starts to bind with different parts of the intracellular compartments. Now, first of all, we can decide that this part right here is known as the sarcoplasmic reticulum, and you should know that in itself, it's a specialized structure within the actual cell, right? And that has a lot of calcium inside, just FYI. Okay, but that membrane of the sarcoplasmic reticulum separates the components inside the sarcoplasmic reticulum from the outside. And so you have a lot of positive cations in there, a lot of calcium. I'll digress. So outside of the sarcoplasmic reticulum, again, you have a lot of positive ions, and they start binding to the troponin proteins on the actual sarcomere. Now, these right here, of course, this whole structure right here is a sarcomere, and let's pretend that these little red dots right here are calcium. And what they do is they start binding to the calcium troponin, the TC, the calcium troponin, or the troponin calcium proteins. And that starts its mechanism where as the calcium activates the troponin protein, it starts opening different channels within the actual sarcomere. And the troponin, what's it called? The tropomyosin, which is another protein. It's this one right here that the TT is pointing at. This tropomyosin, what it's doing, as you guys can see, it's preventing the myosin heads from actually latching on to the actual actin. And the actin is, by the way, T1. And that's this part right here, all these little cups that are looking upside down, that's your actin, right? Another protein. Well, the tropomyosin prevents the myosin heads from attaching to it, right? But once that calcium troponin protein is activated by calcium, it causes the tropomyosin to open up, and this part goes this way. Okay, and let me see if I could just erase it real quick. And now that the tropomyosin has successfully opened the gate, it allows the myosin, this whole part right here, by the way, guys, is the myosin. The myosin heads to couple with the actin, and now we have the actual contraction. Okay, folks, so that whole process is the depolarization phase. Now, it still continues though, right? And by the way, I just didn't draw it, but the whole sarcomere unit does this, and that's just one contraction. The muscle gets shorter, so this part on the lateral end goes this way, and this part gets that way, and that's how the actual cardiomyocytes contract. Now, once that happens, you have a high abundance of a lot of positive calcium inside the cell. During this phase, what ends up happening is, as the calcium channel brings the calcium channels are bringing the calcium inside the cell. Well, you have other channels right here in red. These are your potassium channels, and your potassium channels allow the potassium to leave the cell. Now, this is important because while that's happening, while the calcium channels from the contractile cell membrane allow the calcium to go inside, which is depolarizing the cell, well, your potassium channels are allowing the actual potassium to leave the cell. So that creates that plateau that we discussed previously in regard to the contraction of the actual heart. Okay, now once that occurs, the plateau is there, and then you have that's the depolarization. That's where it ends, and then you have repolarization. Now, repolarization occurs a little bit different. You have a lot of calcium positive ions inside the cell, right? We agreed on that. Well, there's different membranes. Okay, we have different channels right here. This one right here is called the R2R not membrane, but channel, which what it does is, as calcium binds to the protein on the R2R, it opens up the channel and it allows for the highly concentrated calcium that's inside the sarcoplasmic reticulum to come out, and that stimulates other channels that require ATP, because now we have an opposing gradient. Or you know how like diffusion is, ions move from a highly concentrated area to a low concentrated area. Well, in this case, it's the opposite. You have a low concentrated area because you have a lot of calcium inside the sarcoplasmic reticulum. We have to push it back inside. Now, how do we do that? Well, the calcium opens up these specific channels, including sodium calcium channels, and they exchange sodium for calcium. And as they start doing that, we have other membranes on the wall membrane of the cell that allow for more exchange of calcium and sodium. And when that happens, the gates close and the cell becomes less positive inside, and then that's where you have your repolarization. At the same time, in the nodular cells, that potassium channel that I was discussing also opens like this one, and that allows the potassium that's inside the cell to leave it, and that's how it loses its positivity, and it closes the other calcium channels, of course, and it becomes more negative, and that's the repolarization phase. I hope that makes sense. Okay, let me go on to this part right here. Now, this is what's happening during the actual action potential, right? So the nodular cell, the nodular cells have like a non-consistent resting membrane potential, but let's say it's about 160, right? And that's your 160. Once that those funny channels that I was discussing, once they start allowing the influx of sodium coming in, well, the action potential starts increasing, meaning that the cell starts becoming more positive on the inside of the cell. And so that allows, let me do this real quick, that allows the electrical current to start moving upward, to start increasing. Now, once it hits about minus 40, that's what we call our threshold. At that point, that's when that it switches off from, if you guys recall previously, when you have a resting membrane potential, that's when the funny channels open up and they allow sodium to go in, right? So the sodium starts increasing the polarity of the cell. It becomes more positive, and then it goes to minus 55. But once it hits minus 40, well, and minus 55, excuse me, that's when your slow calcium channels are opening, which are bringing calcium inside the cell, makes it more positive. It hits minus 55. But once it hits minus 40, that's when your L channels open and they bring in all that calcium. And that's when they make the cell become a lot more polarized. And that's the depolarization phase. Once it hits positive 40, we're done with the depolarization. And then the potassium channels therefore open and the other positive cation channels close, and that's when you have the repolarization. And then, of course, it hits to its relative resting membrane. That's the action potential for the nodal cells. And in the contractile cells, that's the one that has the phases, the 4 or 5 phases. So we go like this. It's 1, 2, 3, 4, 5 phases. So the resting membrane potential of the contractile cell is consistent with minus 90. The threshold is minus 70. So once you have the once those calcium ions are shifted from the nodal cell to the contractile cell via the gap junctions, well, that's when the polarization starts to increase. You have the resting membrane of minus 90, which is phase 4. Then it starts becoming more positive. Once it hits that threshold of minus 70, as the more calcium starts coming, the sodium and the calcium starts going inside the contractile cell, well, at that point, the depolarization occurs and it hits plus 10. Once it hits plus 10, though, something happens. You have different gating mechanisms that we discussed I just discussed a little while ago, that allow some of the potassium, some of the calcium to start leaving the actual cell, but it plateaus in that segment where the calcium channels are bringing in calcium, but the potassium is letting it out. So even though you're losing some of that potassium, you're bringing in some of that calcium. So it has that relative balance, and that's the plateau phase. That's the second phase. Once that occurs, the potassium leaves the cell, and then we talk about the sarcoplasmic reticulum and how the calcium that's polarized in this phase binds to those proteins on the membrane of the sarcoplasmic reticulum, and it allows the exchange of calcium. Well, that loses the positive ion concentration of the intracellular compartments, and that's what causes the depolarization. And then you have the resting membrane, or phase 4 of minus 90 all over again. And that's essentially how it works, guys. So I hope that made sense. Let me know, and I'll talk to you guys soon.